Good morning, everyone. I welcome you all in this session, which is for AI for reducing clinical trial cost and improving drug safety. There was a bit of delay. I'm really sorry for that. And now we are going to begin with the session. And I hope you are able to see the screen as well. I'm sharing the screen, and let me know if you are able to see the screen. All right. So this session can be attended by students of pharmacy. Along with that, this session can be attended by any other student or professional who would like to understand artificial intelligence role in clinical trial costs and also in improving the drug safety. We will be using some technology here to help us understand that how artificial intelligence will help us in clinical trial as well as drug safety. First of all, we will have a brief about what is clinical trial and what is the role of artificial intelligence in pharma. This is the chapter one, so we will begin with that. Clinical trials. A clinical trial is a rigorously designed scientific study conducted in human participants to evaluate safety and efficacy of medical interventions, drugs, biologics, devices, or diagnostics. The global regulatory framework led by the International Council for Harmonization (ICH) and guidelines such as ICH E6R2 for good clinical practice (GCP) mandates a structured, phased approach to drug development before any medicine reaches patients. You must be remembering the COVID period. During COVID, the vaccination which got evolved. So in India, that was the largest campaign which happened for any such clinical trials. Clinical trial happen for developing a drug. Before the drug is consumed by or used by human beings, it needs to go under multiple phases for acceptance. And we will understand that what are these phases as well. Clinical trials are carefully designed, reviewed, and completed and need to be approved before they can start. People of all ages can take part in clinical trials, including children. There are ethical principles. So all the trials, they must follow the Declaration of Helsinki ICH GCP guidelines and require informed consent from every participant. In India, trials are regulated by CDSCO and must be registered on CTRI. Let us go to this link. So you can see here under National Institute of Medical Statistics, Indian Council of Medical Research. If you want to know that how anyone can get registered into the for as a volunteer for a clinical trial, you can go through this content. Now let me go back. There are four phases of clinical trials: phase 1, 2, 3, and 4. Most investigational treatments must pass through several stages of research before they can approved for use. These stages are called as phases. The phase 1, it evaluates safety and determines appropriate dosage where the drug is first administered to human participants. Phase 2, testing effectiveness and side effects. Phase 3, confirming efficacy and comparing to standard treatments. Phase 4, tracking long-term effects after approval. Let us have a look at. So in case of phase 1, we have a fewer group. The group could be 20 to probably 100 of volunteers. And for them, The primary objective is the treatment safe? Is the drug consumed or drug applied safe? That is the primary objective. Then comes phase 2. Here, the objective is, does the treatment work? So it goes beyond 100, several hundred people, volunteers, they get the treatment. And here, does the treatment work? Is the primary objective. In phase 3, is it better than what's already available? So, for a particular treatment, the drug which is being developed, is it better than a drug which is already available and used by the people? And then phase 4 comes. Phase 4, here the drug is marketed and people can buy it from any pharmacy store. Now, what else do we need to know? Which basically means that when different people are using the drug, phase 4 continuously runs and tries to capture the data that how the drug is actually working or affecting the people. Now, let us parallelly open VS Code where we will refer to, all right, okay. So we have seen that there are 4 phases. Let us have an understanding of these phases in detail. So here you can see that in the phase 1, which is about safety and dosage, first in human, 20 to 100 healthy volunteers are taken. The duration is 1 year to 2 years. The primary goal is determine the drug safety profile, tolerability, and maximum tolerated dose, that is MTD, and pharmacokinetic properties. The key outcomes which come here, that is dose limiting toxicities, that is DLTs, recommended phase 2 dose, RP2D. The success rate is approximately 63% advanced to phase 2. What is the role of AI here? Dose finding algorithms that predict optimal dosing window, real-time safety signal monitoring using wearable sensors, and Bayesian optimization models to adaptively update dose escalation decisions. Understand that from phase 1, it takes 1 to 2 years. That's a big time. When we go for phase 2, it works for efficacy and side effects, and 100 to 500 patients with target disease, they are the registered participants. Duration goes for 2 years to 3 years, and the primary goal is to gather preliminary evidence of efficacy, further characterize safety and dosing. Design types are randomized controlled trials and dose ranging studies. The success rate is 31% of drugs that enter phase 2 advance to phase 3. What is the role of AI here? Biomarker identification for patient satisfaction, predictive models for endpoint achievement, and early futility analysis to halt non-performing arms. Phase 3 is large-scale validation, where 1,000 to 10,000 patients across multiple sites and geographies take part. The duration is 3 to 5 years. The primary goal is confirm efficacy and safety versus standard of care or placebo. It support regulatory submission. Now, the design is typically double blind, placebo controlled, and multi center RCTs. The success rate here is 58% of phase 3 trial results in regulatory approval. This is the most expensive phase. The costing goes up to $300 million to $1 billion plus per trial. It is also where the most late stage failures occur and making it the phase with highest financial risk. What is AI doing here? Predicting primary endpoint outcomes, identifying subgroup population that respond best, automating monitoring through risk-based monitoring (RBM) platforms. Then comes phase 4. In phase 4, it is post-marketing surveillance, where the population is general patient population. So it is thousands to millions wherever the drug is being prescribed. Duration is ongoing for the product life cycle. It continuously goes on. Primary goal is to monitor long-term safety and effectiveness in real-world conditions. Detect rare adverse events not seen in trials. Regulatory obligations are mandated by FDA, EMA, and other agencies. What is the role of AI here? Natural language processing, NLP, on social media, electronic health records, and spontaneous safety reports to detect emerging safety signals at population scale. So you can see that we have understood that what exactly is clinical trials and the four phases in the clinical trials. Now let us try to understand the key challenges. So key challenges in clinical trials. The pharmaceutical industry invests an average of $2.6 billion and 10 to 5 years to bring a single drug to market. And despite this enormous investment, only about 12% of the drugs that enter clinical trials receive approval. You can imagine, just try to understand the cost which is getting involved, the number of years, 10 to 15 years, and only 12% of the drugs that enter clinical trial, they receive the approval. So why is foundational understanding why is foundational to appreciating what AI can fix. So let's understand. So high cost and long timeline. Let us try to understand the breakdown approximately. So we have categories here, that patient recruitment and retention. The estimated cost in percentages, it is 30 to 35%. Site operations and monitoring, 20 to 25%. Clinical data management, 15 to 20%. Regulatory and medical affair, 10 to 15%. Protocol amendments, 10 to 15%. So these are the categories and the cost which is getting involved. Now, why the timeline stretches? Protocol design cycles requiring multiple internal reviews. Regulatory authority review periods often 30 to 90 days for amendments. Site selection, contracting, and startup 6 to 12 months on average. Slow patient recruitment. That's the primary cause of the delays. Data lock and statistical analysis preparation. What is the industry benchmark? The average phase 3 trial takes 7.5 years from IND filing to NDA submission. IND and NDA are the processes. So every day of delay in drug development costs a sponsor approximately $600,000 to $8 million lost in revenue, depending on the drug's market potential. So understand the loss as well. for the sponsor. Sponsor is that company which is sponsoring the trials. So it requires sponsors as well because it's a huge cost. So patient recruitment delays. This is a single greatest operational challenge in clinical trial. Key statistics, you can see 86% of the trials fail to meet enrollment deadlines. 19% of the trials fail entirely due to insufficient enrollment. The average patient takes 21 months to enroll in a trial after diagnosis. What is the root cause of it? Over restrictive inclusion exclusion criteria that eliminate 90% of the potentially eligible patients. Physician unaware of the open trials, geographic barriers, patient far from the trial site, patient's hesitancy, that is another challenge. Then concerns about placebo, randomization, and side effects, and competing trials recruiting from the same patient population. So, what are the cascading effects? A 1 month delay in enrollment adds 600,000 to $3 million in trial cost due to extended site operations, staff costs, and delayed market entry. Now, another point is trial failure and inefficiencies. Failure categories are scientific failures, operational failures, strategic failures, and safety failures. Now, of every of every 10,000 molecules screened, only one reaches the market approval. Of every 10,000 molecules, only one reaches the market approval, and the transition success rates. So, preclinical to phase 1, 10%, phase 1 to phase 2, 63%, phase 2 to phase 3, 31%, phase 3 to approval, 58%, overall phase 1 to approval, 12%. Operational efficiencies often include manual data entry leading to transcription errors, duplicate data collection across systems, inefficient site monitoring visits, many are not needed, protocol amendments invalidate months of work, that invalidate months of work, and then delays in safety data review and escalation. Now, what is the role of AI and ML in modern drug discovery? The role of AI and ML. So, artificial intelligence in pharma is not a futuristic concept. If you try to go to website of any pharma company, you will find the sections of where what research and how AI ML capabilities they are having. So artificial intelligence is not a futuristic concept. It is operational today at leading organizations including Pfizer, Novartis, Roche, AstraZeneca, and many AI biotech like Recursion Pharmaceuticals, Insilio Medicine, and Benevolent AI. So where AI creates value? First, drug discovery. Generative AI designs novel molecular structures with desired pharmacological properties. Deep learning predicts protein folding, AlphaFold, which is released by DeepMind, has solved structures for 200 million proteins. ML model predicts ADMET, absorption, distribution, metabolism, excretion, toxicity, properties computationally replacing many animal studies. Then clinical trial design. AI generates optimized protocols based on historical trial data. Bayesian adaptive designs adjust trial parameters in real time. Simulation models predict enrollment timelines before a trial starts. Patient recruitment, NLP, natural language processing, scans EHRs, electronic health records to identify eligible patients. Machine learning scores sites by their historical enrollment performance. Digital patient machine matching platforms, Antidote, Trial Spark, connect patient to trials. Then data monitoring and safety. Risk-based monitoring, RBM. Systems prioritize which sites need on-site visits versus remote review. Then real-time safety dashboards aggregate adverse event data across sites globally. NLP processes free text and safety narratives at scale. Then we have regulatory submissions. AI automates portions of clinical study report writing, which otherwise will is already taking a huge time. Natural language generation tools generate, they can draft sections for regulatory documents. Semantic search tools help medical writers find precedent in prior submissions. So, this is how AI is contributing in clinical trials. So, the AI value chain in clinical trial will look like data ingestion, data cleaning, feature engineering, model training, then prediction insight, decision support system, clinical action, and outcome measurement. Next is evolution from traditional to AI enabled trials. So, in evolution from traditional trials to AI enabled trials could look like traditional trial paradigm pre 2010, paper based case reports, CRFs, then periodic site monitoring visits, 100% data review, static protocol design, no mid trial adjustments, siloed data systems, EDC, CTMS, safety databases rarely integrated, statistical analysis only after database lock. Then the transition period from 2010 to 2018. So electronic data capture systems became standard, EDCs. Electronic clinical outcome assessments, ECOA, electronic clinical outcome assessments. They reduced site burden. Risk-based monitoring introduced FDA guidance in 2013. Risk-based monitoring. First user of predictive analytics for enrollment forecasting. It came between 2010 to 2018. Introduction of variables in trials like accelerometers for activity data. Now, 2018 to present, AI-enabled trial paradigm. Real-time data streaming from multiple sources. Electronic health records, variables, labs. Adaptive trial design informed by interim analysis using ML. AI-powered patient recruitment platforms. NLP-driven safety signal detection. Digital twins allowing simulation before first patient in. Decentralized clinical trials, DCTs, enabled by remote monitoring and telemedicine. So, from traditional to AI-enabled trial, there has been a transition phase, and that shift has come, and now it is operational, it is being done. Real-world data and digital transformation in pharma. So real-world data is data collected outside of traditional randomized clinical trials from the actual clinical care of patients. It can include the EHR's electronic health records. They are structured and unstructured patient data from hospital systems. Then claims and billing data, insurance claims, capturing diagnosis, procedures, and medications. Patient registries, disease-specific observational data, patient registries. Then wearables and mobile health data, which comes from continuous vitals activity like sleep, glucose monitoring. Then social media and patient forums. So patient reported experiences and symptoms through social media and other forums. So real world evidence, when RWD is analyzed systematically to generate clinical evidence, it becomes real world evidence. The FDA's 21st Century Cures Act 2016 formalized the use of real world evidence RWE in the regulatory decisions, including supporting new indications for drug approval, satisfying post market study requirements, informing trial design example using RWD as external control arm. Digital transformation drivers in pharma, so cloud computing, interoperability standards, patient centric technologies, decentralized clinical trials, and AI platforms. So, with this, the convergence of AI, RWD, and digital technology is not incrementally improving clinical trials, it is fundamentally redesigning them. For young professionals, this means that domain expertise in clinical operations combined with data literacy is an exceptionally powerful combination. So, with this, we end the chapter 1, where we discuss the concept of trial phases, core challenges. So, core challenges 88% failure rate, 2.68 per approved, okay, $1 billion per approved drug, very high cost. Then AI entry points, discovery, design, recruitment, monitoring, and pharmacovigilance. Here AI can get entered and can support. RWD, the real world data. So data from routine care increasingly accepted by the regulators because they are the decision makers. Digital transformation, DCTs, wearables, electronic health record integration, AI platform reshaping the trials. Okay, some discussion questions, thoughts to ponder. So, if you are designing a phase 2 oncology trial, which three AI use cases would you prioritize and why? How does the 12% overall success rate of clinical trial justify the industry's investment in AI? What ethical concerns arise when using real world data in regulatory submissions? Now let us go to the chapter 2, that is understanding clinical trial cost drivers. This will help us to understand that why like we are here discussing this particular part, that is understanding, that is for clinical trial cost. So the clinical trial cost drivers, what we will try to understand in this particular chapter: identify and quantify the major cost component of a clinical trial, understand how operational failures escalate costs, ex- explain the financial impact of trial delays and protocol amendments, describe specific AI applications that reduce primary cost. Yes, so we will be covering these particular part. Okay, fine. So the major cost components, what are the major cost components involved? To apply effective AI, you must first understand where money is spent and where it is lost. Clinical trial costs have been risen by over 100% in last decade, driven by increasing protocol complexity, regulatory requirements, and the growing cost of patient recruitment. The full cost stack for a phase 3 trial. So here you can see patient recruitment and retention. The major cost which goes is patient recruitment and retention. That's the major cost where it goes. Yeah. So here if you see, let me just adjust something. Yeah. Okay, fine. So Cost impact is 30 to 35% of the total trial budget for patient recruitment and retention. Now, patient recruitment is consistently the most expensive and most challenging operational component of a clinical trial. What are the sub components if you see? So, screening cost. Each screened patient who fails eligibility, that is screening failure, cost 1,000 to $15,000 depending on the disease area. Screening failure rate in complex disease like autoimmune conditions or rare cancers, screen failures rates can exceed 70 to 80%, meaning you pay to evaluate 5 patients for every one enrolled. Okay, so you pay for evaluate 5 patients for every one enrolled patient. Now, investigator site fees. So sites charge per patient fees for enrollment, then follow-up visits and procedures. Digital recruitment campaigns, social media, online patient community outreach, search engine advertising. So that is digital recruitment campaign. Then patient navigation programs, coordinators who help patient navigate logistics like to travel, insurance pre-authorization, retention and adherence programs like patient who drop out after enrollment create missing data, which inflates the sample size requirements and the cost as well. So cost of screen failures, if a trial need 500 enrolled patient and has a 75% screen failure rate, you must screen 2000 patients at $3,000 per screen failure and $5,000 per enrolled patient. The recruitment cost alone reaches $7.5 million or above before any treatment phase costs. So what is the AI opportunity here? This this data helps us to understand the depth that there is a requirement to improve, there is a requirement to optimize. So AI opportunity is machine learning models trained on historical EHR data can predict which patients are likely to pass eligibility criteria before any screening visit, dramatically reduce screening failure rates. Companies like Medidata, Viva, Trinetics provide such predictive enrollment tools they are providing. Now, site selection and operation. So, cost impact is 20 to 25%, 20 to 25% of the total trial budget. A multinational phase 3 trial may run across 200 to 500 clinical sites in 30 to 50 countries. Site selection and ongoing site management are enormously resource intensive. So site selection cost, feasibility, feasibility questionnaires sent to hundreds of candidates, site qualification visits, SQVs to assess infrastructure, contract and budget negotiation, legal and finance teams do that. IRB and ethics committee, the regulatory bodies, committee submission at each site, and site initiation visits, SIVs, to train the site staff. Then operational cost, this is the selection cost. Then operational cost, site operational cost, it involves clinical research associate, that is CRA, time for monitoring visits. Then source data verification, SDV, reviewing 100% of the source data is traditional standard but very costly. Data query management, resolving data discrepancies. Then site management team oversight, site payments, invoicing and reconciliation. The under enrollment problem, 30 to 40% of the trial site enroll fewer than 3 patients in the entire trial. 30 to 40% of the trial site they enroll fewer than 3 patients. These low enrolling sites still receive full site setup cost, which is often $50,000 to $200,000 per site, and ongoing management overhead without contributing meaningful data. So this one of the most wasteful areas, okay, in clinical operations. What is the AI opportunity here? So AI models trained on historical site performance data from internal database and also public registries like clinicaltrials.gov. It can predict a site likely enrollment yield before commitments. So this will allow the sponsors to focus resources on high performing sites. Next part is data management and monitoring. The cost impact goes 15% to 20% of the total trial budget. Data management cost, EDC, electronic data capture system licensing like Medidata Rave, Oracle Inform, Viva Vault, database design, testing, and validation. So per 21 CFR Part 11. Data cleaning, query management, and resolution. Medical coding, MedDRA for adverse events, WHO drug dictionary for medications. Statistical programming like SAS, R for analysis data set. Then clinical study reports, CSRs, writing and review. Then comes the monitoring cost. Monitoring cost is traditional 100% source data verification. It requires CRAs to visit sites frequently and often monthly for fast enrolling sites. Then the Tufts Centers for the Study of Drug Development estimates that monitoring accounts for 25 to 30% of the clinical operation costs. FDA guidance 2013 and ECH 6R2 2016 both endorse risk-based monitoring as modern standard. Now, the shift to risk-based monitoring. Under the RBM, that is risk-based monitoring, AI and statistical systems continuously analyze incoming data to identify sites with unusually low or high adverse event rates, potential reporting issues, data patterns inconsistent with disease expectations, and protocol deviation trends suggesting training gaps. So this allows monitoring resources to be directed where risk is highest rather than applied uniformly. Now, the other section is 2.2 is operational inefficiencies and failure points. Understanding where the trials operationally break down is an important as important as understanding the cost, because operational failures often generate the largest unexpected cost. So far what we were discussing, it was more on the expected cost, like what is the expected cost which is going on. Now the operational efficiencies, this you can understand with a simple example. Let's say you planned a trip and each and every activity in the trip is time bound. Let's say you stayed in a hotel. In the morning at 8 a.m. the vehicle is supposed to come, but none of the group members are ready at 8 a.m. So they basically woke up at 10 and got ready by 12 and then went for the breakfast. Now understand, let's say there was a free breakfast offered, it is gone. The vehicle which was arranged, it is gone. Now, because of operational inefficiencies, the breakfast cost as well as the transport cost will be additionally charged now. And it is not under the package specifically for your group, it has to be charged. So I hope this makes the analogy, this way you can understand. So understanding where trials operationally break down is important. Now, common operational failures, protocol design failures, eligibility criteria so narrow that too few patients exist, endpoints that are impractical to collect, 10-year survival in 3-year trial. Okay, 10-year survival in a 3-year trial. Visit schedules that are burdensome to patients and increase dropout. Inadequate sample size calculations leading to underpowered trial. Then site performance failures. Investigator turnover mid-trial requiring retraining. Then inadequate site staff for the data entry or visit coordination. Poor patient population in catchment area. Completing trial recruiting the same patients at the same site. Now, data quality failures. High query rates. More than 10% of the data fields queries indicate systematic problems. Protocol deviations in key efficiency, efficacy assessment. Missing primary endpoint data can invalidate an entire trial. Biggest loss: unreliable biomarker or laboratory data. Then regulatory failures are there. So protocol amendments required by the health authorities mid-trial, clinical hold like FDA or clinical trial suspension like EMA due to safety signals. Then GCP inspection findings that require data remonitoring. So understand the quantifying effect of the impact. A single unplanned protocol amendment cost an average of 450,000 dollar in direct cost and adds 3 to 6 months to timeline. A phase 3 trial that goes on clinical hold can lose 1 million to 5 million dollars per week while the hold is in effect. Next section is impact of the trial delays and protocol amendments. Okay, and then we will go for the AI use cases after that. So, the economics of delay. The pharmaceutical industry operates under time limited patient protection. Once a drug is approved, the sponsor has a defined period, typically 7 to 12 years remaining post approval, to recoup development cost and generate profit before generic competition enters. So, cost of a 1 month delay, phase 3, direct operational cost 1 to 3 million dollar for site operations staff, investigational product management. Opportunity cost loss. So opportunity cost is lost sales. 600,000 to 8 million dollar depending on the drug's market potential. Then competitive risk. Competitors may reach market first. That's another risk. So protocol amendments. So according to the Tufts CSDD, 90% of the phase 2 and 3 trials undergo at least one unplanned protocol amendment, and the average complex trial has 3.5 amendments. So that's another thing which is adding to the cost. Then amendment cost drivers are regulatory authorities. The regulatory authorities feedback requiring design changes, safety signals requiring close adjustments or additional monitoring, poor interim results suggesting subgroup restratification, and operational problems. Example, a primary endpoint that is not being collected correctly. So the cumulative financial impact in a large phase 3 program, protocol amendments have been estimated to cost around $100 million to $300 million cumulatively across all amendments and their cascading effects on lifetime, sample size, and site operations. Now, AI use cases for cost reduction. Let us try to understand this part. So predicting patient enrollment rates. So after this, we will see one demo as well. Okay, so predicting the patient enrollment rate, the problem is most enrollment forecasts are based on site investigators, self-reported estimates, which are notoriously optimistic. Industry data shows actual enrollment is typically 50% of forecasted enrollment. 50% of forecasted enrollment. So the AI solution would be data inputs used by ML enrollment prediction models, historical enrollment data from same therapeutic area, site site level performance metrics from prior trials, disease prevalence data in the site's catchment area, clinicaltrials.gov data on competing trials recruiting the same population, EHR derived patient population estimates. Now, what kind of models will be used? Regression models will be used. Predict monthly enrollments rates per site. Regression models, they can predict monthly enrollment rates per site. Then Monte Carlo simulation. It will generate probabilistic enrollment timelines 10th, 50th, 90th percentile scenario. Then time series forecasting should be used. Adjust as actual enrollment data comes in. Okay. So example AI outcome could be, this is just an example. So a global oncology trial using AI enrollment prediction identified 12 sites out of 80 candidates that were predicted to enroll fewer than two patients each. By excluding these sites and redistributing the budget to eight higher performing sites, the sponsor reduced the startup cost by $3.2 million and met the enrollment target 4 months ahead of schedule. This can be the impact of AI being integrated. The second part is optimizing trials site selection. The problem statement is traditional site selection relies on investigator relationships, reputation, and feasibility surveys, a process that is subjective and slow. Typically, it takes 6 to 9 months. The AI solution is a machine learning site selection model will consider historical enrollment rates for same protocols, site infrastructure, dedicated coordinator FTEs, patient databases, geographic proximity to target patient populations, regulatory track record, prior inspection findings, deviation history, then competitive trial burden, sites running many competing studies, site financial performance like invoicing accuracy, compliance with the budgets. What type of techniques can we use? We can use random forest gradient boosting. Random forest and gradient boosting will help ranking the sites predicted enrollment yield. Okay, so by predicted enrollment yield, it will rank the site. Then clustering algorithms group the site performance by profile to diversify the site network. And then NLP, natural language processing, extract relevant site attributes from feasibility survey free text responses. Okay, so some relevant site attributes can be extracted and they can be processed through the natural language processing. Now, industry tools which are there. So we have Medidata Site Connect, IQVIA Site Intelligence, then Anti.me, Viva Vault, CTMS with embedded analytics. So these are the current tools which are available. Case discussions, cost reduction using AI driven trial design. So, oncology phase 3 trial redesign at a major pharma company. Let us understand the background. A major pharmaceutical company was preparing a phase 3 trial for a solid tumor indication. Initial protocol design projected 1200 patients enrolled across 150 sites in 25 countries. Enrollment period 36 months. Total trial budget $480 million. Timeline to database lock 5.5 years from first patient in FBI. Now, what are the challenges identified? The challenges identified: historical screen failure rate in this indication 72%. Prior phase 2 data suggested a strong responder subgroup, approximately 40% of the patient. Then 35% of the planned sites had enrolled zero patient in similar prior trial. These are the challenges identified. Now, how AI interventions applied and can help. So here if you see biomarker driven eligibility redesign, and an ML model trained on phase 2 genomic data identified a biomarker signature that distinguished responders from non-responders with 78% accuracy. The team redesigned eligibility criteria to enrich for the responder population. This reduced the required sample size from 1200 to 680 patients. Okay. Then, second is AI powered site selection. So, a boosting model scored 350 candidate sites on 22 features. The top 90 sites were selected. Top 90 sites. 35 sites predicted to enroll zero patients were excluded. It saved $12 million in site startup cost. Another point, enrollment forecasting with Monte Carlo simulation. The simulation showed 85% probability of meeting enrollment with 90 sites in 22 countries versus 150 sites in 25 countries originally planned. Country selection was optimized to favor regulatory environments with fast site initiation. Fourth one is risk-based monitoring implementation. AI-driven risk indicators reduced required on-site monitoring visits by 40%, saving $18 million in CRA travel and operation cost. So the outcome achieved are sample size, original plan 1200, AI optimized plan 680, saving 43% of reduction. Number of sites originally 150, AI optimized 90, 40% reduction. Enrollment period 36, AI optimized plan 26 months and 14 months faster. Total budget $480 million. AI optimized plan $295 million, $185 million saved. Timeline to DBL 5.5 and here 3.8 years under AI optimization plan, 1.7 years faster. So the key lessons are the most powerful AI driven cost reduction. Okay, come not from automating the existing process, but from fundamentally redesigning the trial with AI insights before the first patient was enrolled. So there is a complete shift. There is a complete shift in the process. So this is the critical message for young professionals, and the best time to apply AI to a clinical trial is during the protocol design phase, not after the trial start, but during the protocol design phase. So the chapter 2 summary says that recruitment cost 30 to 35% of the budget, screen failures are major waste driver. Site operation 30 to 40% of sites enroll fewer than 3 patients, highly inefficient. Protocol amendments 90% of trials have at least one. Average cost 450,000 each. Then trials delay. Every month of delay cost 1 to $8 million depending upon the drug value. AI for enrollment, ML models reduce screen failures, improve site selection. AI for monitoring, RBM reduces monitoring cost 25 to 40%. Now, chapter 3, AI ML fundamentals for clinical trials. So before we enter into this, we will first try to have a look at uh, mini project or a demo project which we can work with, which we can cover. So let me introduce and I will help you with that how you can implement it. So first of all, we require VS Code. This particular screen itself shows that this is not something notepad kind of thing. Rather, this is an IDE. IDE basically means that where you can write codes and you can develop your projects. So this is VS Code, where I have opened this particular file, and we are exploring through that. So you can install this VS Code. How you can install VS Code? You need to go to the Microsoft Store. So you can go to Microsoft Store, and there you need to search for VS Code. So here VS, so you'll find this way. So in my case, Visual Studio Code is already installed. It is already set up. So I need not to install it, but you will have to install it. And from here you can do it. The other option you have is you can install it from downloading it from official website as well. So two options you have. It is very simple to use IDE. Now, once you have done this, then the next thing is you can open it. You can just type VS Code and click on it. It will get opened. So I'm opening the second window of it, where I can help you to walk through. So this is Visual Studio Code. And here, if you want to work with a project, then in that particular case, you need to work. You need to have familiarity with the interface. So this interface, you can have a look at it. Okay. So in this interface, you can see that we have file, edit, selection, view, go, run, terminal, help. Then we have options here: Explorer, search, source control, run and debug, extensions, testing, metals, Azure, AI Toolkit, Microsoft Foundry. So we have these multiple options here. Initially, you can create or you can select a project directory. That you can do from here, open folder. So once you click on open folder, here you can find that either on desktop you can go and do a right click, go to new, and you can click on folder, and here you can write project project name. So project 1, enter, and then select folder. So if you do this in this particular case, you will find that it will ask you whether you want to trust the authors. You have to say yes, and it will open that particular folder as a project. Here you can see. Fine. Now, under this, you need to create your project files. This is step number one, Visual Studio Code. Okay. So if you are doing it parallelly, you may pause the video. You can go to Microsoft Store and you can install it from there. Okay, you can do it like that. Once you have installed it from there, then after that, okay, you can come back and follow the video. Now let me go to, yes. Okay, so look at this. Okay, fine. So here you can see the objective which we have planned. It is that clinical trial AI dashboard. And with this particular objective, we want to have end-to-end AI-powered decision support system for clinical trials. This helps pharma teams predict trial cost, predict adverse event risk, and understand key drivers, that is explainability in AI, and optionally it will generate AI insights using LLMs. The core objective is the system solves two problems in clinical trials. The first one is cost prediction. It uses ML, machine learning, to estimate total trial cost. It helps to optimize budget planning. Then risk prediction. So predicts probability of high adverse events, helps improve drug safety. So we have two objectives, one for cost prediction and second one is the risk prediction. Now, for this, we need a data set. The data set has a couple of features. It is named as clinical trial cleaned dataset.csv. It is the data set. And these are the features inside it. Phase, the values inside the phase could be 1, 2, 3, 4. So that is first part. Then sample size, number of sites, countries, visits per patient, disease area, baseline AE rate, dropout rate, recruitment rate. Then we have target variables also. Target variables are cost, which is a regression target. Regression target means that the cost will be cost is a continuous variable, and therefore the prediction for cost will be coming from a regression approach. Then high AE risk. AE is adverse event. So high AE risk, it is going to come from classification target. It is a classification parameter. So it will be coming from classification. Then next part is workflow pipeline. So here let us have a look at the other files. So you can see that we have we have this clinical trial cleaned data.csv. This file you can see we have. And in this file, you can see we have phase, sample size, number of sites, num countries, then visits per patient, double blind, disease area, baseline AE rate, dropout rate, recruitment rate, cost, and high AE risk. This type, this file you may get from some sources, and these sources could be kaggle.com, GitHub, or any open data source platform. So this type of a data you can get from any such yeah so you can get from any such places. Okay, now let us go back. Now second one is workflow pipeline. So workflow pipeline, here we have step number one is data loading. Once you have the data ready with you, after that, the second point is you need to load the data. So loading the data means the model is going to read the data, learn from the data, and then it is going to make the prediction. So this is how it is going to happen. Step number two is pre-processing. So once the data gets loaded, next step is pre-processing the data, which means that making the data ready for the model to learn from. In pre-processing, there are many things, but this data itself is cleaned data. So we require one pre-processing step, that is converting the categorical data into numeric. So how does that happen? Like converting the categorical data into numeric. So it happens with the help of pd.getDummies. So there is a function which does it. It converts the categorical data into numerical. And this technique is called as one-hot encoding. Once we have the data present in the form of numbers or numeric format, then next step is feature split. Feature split is that data needs to be in the form of input and output. We have two output features. One is Y cost and the second one is Y risk. Y cost is cost prediction, Y risk is risk prediction. And then rest of the X or input features, they will become, they will be the input features. This is how we will split the features. Now further, the next step is train test split. So this step number 4, which is train test split, it is used for dividing the data into training and testing. We divide the data into training, validation, and testing. So why do we do so? Because when we divide the data into training and testing, at that particular time, the model is trained on the training data, and then model is evaluated using the testing data. So this way you get the evaluation also done for the model. This way evaluation is also done. If you have validation data as well, in that particular case, before you go for evaluation, you validate as well. When you validate, then you are assured that the training has happened in a right manner. So therefore, train test split is being used, where you are splitting the data in the form of training and testing, or in the form of training, testing, and also validation. Now, the next part is machine learning models. So, in machine learning models, we have two things. One is, you can see, yeah. So, one is cost model, which is regression model, and second one is the risk model, which is classifier model. So, this particular part, if we want to understand that machine learning has got multiple types. So let us go back here and yes, try to understand this part. So AI ML fundamentals of clinical trials. So for this, we will have to understand this. Yes. So the objective here is categorize the AI ML model types most relevant to clinical trial applications. Then identifying the primary data sources available for clinical trial analytics. Execute standard ML workflow from data ingestion to model evaluation. Apply techniques for handling common data quality challenges in clinical data. So we'll try for that. Okay. Now here if we see the types of AI models which are used in clinical trials, understanding which tool to apply to which problem is foundational skill. And many professionals also struggle on this because it requires conceptual understanding, domain knowledge, and as well as experience. So, here in this case, clinical trial problems span a wide range of tasks. For example, predicting predicting a binary outcome, will this trial succeed, to extracting structured information from unstructured clinical trial narratives, which is a very complex task. Now, in case of machine learning, we have a supervised approach. Supervised learning trains a model on labeled data, which means that examples where correct answer is known and learns to predict the label for new unseen examples. Classification, predicting discrete categories. Under supervised, classification is one type. For example, binary classification examples. Will this patient experience a serious adverse event? What will be the output? The output will be yes or no. Will this trial succeed? The output will be pass or fail. Then is this patient eligible for this trial? The output will be eligible or ineligible. So this is binary class, two class classification. Now further we have is multi-class classification. So in case of multi-class classification, what is the severity of this adverse event? So what is the severity? So it could be mild, moderate, severe, life threatening. So this part, if you see, it is having more than one classes, not just one class, more than one classes it is having. Now, next one is which of the five trial site is this patient best matched to? So this is the here also out of five, you are going to get one output. Okay, so that's again, if you see, is an example of multi-class classification. So multiple classes are there. Okay, now the algorithms which are used here. So we have logistic regression, random forest, gradient boosting, XGBoost, light GBM, then support vector machine, neural networks. These are the five listed algorithms which can be used here. Logistic regression, the strength is interpretable, regulatory friendly. As we know that we need to have regulatory friendly algorithms. So logistic regression is interpretable and it is regulatory friendly also. Then adverse event prediction. Okay. So this is one of the baseline parameter we need to consider when we are designing the the model for predicting the safety or risk, for predicting the risk. Random forest handles non-linearity, robust to noise. So random forest, it handles non-linearity and it is robust to noise. Trial success prediction and site scoring can be done with the help of random forest. Then we have XG gradient boosting. Algorithm name is XG boost or light GBM. High predictive performance, patient enrollment prediction can be done. That's a use case. Then support vector machine, which is SVM, effective in high dimensional space. It is used for biomarker classification. Then neural networks, multi-layer perceptron captures complex patterns, multimodal outcome prediction. Now, these are classification. Now, regression. Regression, another type of supervised only, which is used for predicting the continuous values. Examples in clinical trials, predicting the number of patient a site will enroll over 12 months, then predict a patient's clinical response score, continuous endpoints like SLE, DAI, MMSE, then estimate time to event, time to disease progression. The second type is unsupervised learning. This is another type. Okay, unsupervised learning. So in this particular case, the unsupervised learning, it finds hidden structure in the data because the data is not labeled. It is unlabeled data in this particular case. So it has got techniques like clustering. Clustering is group patient by disease subtype based on genomic or clinical features. So here we are doing group. Okay, so that is basically technique where we do not know or we don't have the prior knowledge. We don't have the prior knowledge of how to classify or how to name. So we group, but we do not know the name. Okay, what name should we give to the group? That we don't know, but we have grouped. Okay, now the next thing is cluster trial sites by performance, profile for monitoring optimization. Okay, so trial sites with the like we can prioritize, then identify anomalous sites or patient. So they are outliers. Anomalous sites we discussed in the previous example where the sites are non-performing. So we can reduce, we can basically not include them because they are incurring very heavy expense. So we can exclude in that particular case. The kinds of algorithms which are used, K-means algorithm, which is simple, scalable, and interpretable. Then clustering, hierarchical clustering, it reveals nested patient subgroups. It is creates hierarchies, and it is again explainable and interpretable. DB scan, it handles irregular cluster shapes. Many times the cluster shapes are not regular or simple. So DB scan is an algorithm, a technique. It handles irregular cluster shapes full of outlier detection. Then another concept is dimensionality reduction. Many a times, if there are lot of parameters and all of these parameters are to be processed by the algorithm, then in that particular case, the algorithm running on a system, the system might face challenges. Let's say you are consuming 1 GB of RAM now, and 1 GB is sufficient for 10 parameters, 10 features. But if you have 200 parameters to be processed, so in case when you have 200 parameters to process, you need to reduce the dimensions to less number, so that if you increase, let's say, from 1 GB to 2 GB, what will happen? If you increase from 1 GB to 2 GB, what is going to happen? Then it might be able to handle 20 parameters, but for 200 parameters, okay, you will require extensive hardware to process that particular data. So in that particular case, we try to reduce the dimensions. So for example, if there is a cost factor which affects which if the cost is getting affected by number of sites, and the cost is also getting affected by the number of patients, so can we design a formula so that the number of patients and the number of sites, we can merge these two values and bring a one column, one feature out of these two. If we are able to do this, this basically means that we are able to reduce the dimensions. Dimensions are number of features, number of parameters one algorithm needs to process in the data set. So the techniques which are used for dimensionality reduction, they are principal component analysis, PCA, principal component analysis. It reduces the high dimensional genomic data to manageable feature set. So it works in this way. It reduces the high genomic data into a manageable, which means low dimensional. It converts it into low dimension. That is how it works. Then the next part we have is deep learning. Okay, so deep learning uses multi-layered neural network capable of learning hierarchical features represented from raw data. Deep learning uses multi-layered neural networks capable of learning hierarchical features representations from raw data. To understand it in simple words, think of your biological neurons. So, biological neurons are cells. These cells receive input, they get activated, and they give some output. The way you touch a very hot plate, so the cells on your fingertips, they will receive the input, they will get activated, and they will transmit this information. Now, very similar way, we create such a network with the help of creating artificial neurons, and that is the area of deep learning, the branch deep learning. The types of neural networks we have are convolutional neural networks, recurrent neural networks, and transformer models. Convolutional neural networks, CNNs. So convolutional neural networks, their purpose is Yeah. So the convolutional neural network, their purpose is to handle the images. They broadly handle images. So these convolutional neural networks, they help in medical image analysis, tumor detection in radiology or pathology slides, clinical trial endpoint ad, yeah, adjudication from imaging data. So MRI in MS trials, it is able to work on imaging data, dermatology imaging for safety assessments. As I said, convolutional neural networks, they essentially work for the images. They take the image, they are able to process the image, and and they are able to bring the they are able to bring the features out of images. Like for humans, it is easy to look at an image and identify the patterns. Patterns like how far is that image, what is the depth of the image, what is the height of the image. So we are able to identify. Similar way, neural networks are also able to identify hundreds of such features to learn from the images. Then we have recurrent neural network. The recurrent neural network, if you see, the purpose of recurrent neural network is it is yeah, this one, the recurrent neural network. It is used for time series data. By time series, I mean that if I say I am suffering from cold, so I am suffering from cold, if machine learning learns from this sentence, so it will be able to learn that if I say I, then the prediction should be am. If I say I am, the prediction should be suffering. So with you can see the sequence, time series basically means a sequence. So with a sequence, how the next item should get predicted. So recurrent neural networks or LSTM, long short-term memory, they work with the time series data, and at the same time they also work with natural language processing. For both the things they work. So time series data from variables like ECG, then accelerometry, then continuous glucose monitoring, and then predicting longitudinal clinical outcomes based on serial lab values, even sequential EHR data analysis. Then we have transformer models. Transformer models are the latest models or something which is very much trending. They come with large language models, LLMs, for clinical text understanding and clinical trial protocol analysis and optimization, medical question answering system. So here the large language models, which are trained on very huge data, and the data comes from that specific domain. So they become expert to understand and respond back the queries related to medical questions. Now, the key considerations for pharma: deep learning models are powerful but require large data sets and careful validation. This is critical for regulatory submission because they have a black box nature. Black box nature is that it doesn't explain everything. So it requires explainability frameworks. It is covered in further chapter we will go through. So deep learning is very powerful, but it requires explainability. And that is the major drawback with such deep learning models. It's a drawback. Now, next thing 3.1.4, that is natural language processing. Okay, natural language processing. So clinical data is estimated to be 80% unstructured. Clinical data is estimated to be 80% unstructured, sitting in free text notes, adverse event narratives, clinical study reports, and scientific literature. Here, NLP unlocks this data. NLP is natural language processing, where human like conversation, human like conversation is happening. So the app, so the NLP pipeline for clinical text, raw text, it is it goes tokenization, part of speech POS tagging, named entity recognition, then relation extraction, then classification, summarization, and then structured output. So the clinical text is considered as raw text. It is split into pieces. It is called as tokenization. When the text is split into pieces, it is called as tokenization. Then part of speech tagging, like in English. So there is part of speech tagging. Then named entity recognition. Named entity. So clinical names, disease, procedures, diagnostics, equipment. So they have names. So named entity recognition help us to identify these medical terminologies. Then relation extraction. How one entity is related to other. Relation extraction. Then classification or summarization. So classification means we have discussed, like classifying diseased, not diseased, healthy, yes, no. So two class, multi-class. So classification or summarization. Summarization means bringing it to like bringing a summary to what has been passed as a text to the what has been passed as a text to the model, and it gives the structured output. So that is the whole pipeline for the clinical text. Now, clinical NLP applications, adverse event processing. Okay, just give me a second. Okay, so adverse event processing, extract structured adverse event information from spontaneous reports. So MED-DRA coding is used for that. Then patient eligibility screening, parse EHR notes to identify trial eligible patients. Protocol analysis, extract inclusion, exclusion criteria, and map to structured formats. Literature mining, identify relevant prior studies to support drug development decisions. Clinical document generation, generate first draft of narratives, summaries, and clinical study reports. So you can see that the clinical ML NLP applications goes wide: adverse event processing, patient eligibility screening, protocol analysis, literature mining, and clinical document generation. What kind of techniques are used here? So the NLP techniques used here are rule-based systems, rule-based systems, which are fast, interpretable, and good for structured extraction. Then we have classical ML, like naive Bayes is used, SVM is used with TF-IDF features, then useful for document classification. Then we have BioBERT, we have clinical BERT, so pre-trained transformer on biomedical text, state of the art for most clinical NLP tasks. Then GPT-4 or Claude is there, general purpose LLMs now being integrated into pharmacovigilance workflows. Now, the next step is data sources for clinical trial AI. But before that, we will go for the project part again. Okay, so we had seen this cleaned data. Okay, we had seen this clean data, and then we went on to discuss on the different models. So we currently had discussed multiple models which help us to work on different areas of clinical trials. And now we are just confirming here that we are using XGBoost for regression, and next one is we have a random forest. Here you can see for classification. So these two we are making use of. Okay, and further what we will do is we will save the model, and model will be saved as cost underscore model dot pkl. Okay, so that is how the model will get saved. Fine. Then we will create a user interface. So here you can see we will create a user interface. For that we have Streamlit, which is one of the Python library. It is being used, and with that we will design the whole UI. So here in VS Code, I should be able to work with a terminal. You can see this is the terminal where I will run this application. So Streamlit run. Okay, so CD. Yeah. So we will run this application, and you can see that this application will run on a browser, local browser. Here you can see. So it is taking some time. Okay. So here you can see. So just have a look at this, and we will continue after a minute. So you can take a short break here of 2 minutes, and then we will continue after that. So we'll understand this dashboard and this how this application works. All right, so let's continue with this dashboard. So here you can see that the data which we had discussed. So that same data is very much visible here, that same data. So the model is going to learn from this data. And after learning from the data, the model can make the predictions. So how the model is going to learn, that also I'll explain. So this is input data. We have shown the same data over here. You can see phase, sample size, num sites, num countries, visits per patient. The same data is here. And then here we have given option of training the model. Then you can give your input, the new input, and then you can go for prediction. Okay, so let us do this. So train the model. You can see models trained successfully. And now you can choose like which phase you are working. Is it phase one? Is it phase two? Is it phase three? So for which phase you are working? So let's say it is for phase two. So you have selected phase two. Now for every phase, you will see for phase one, you can see there is a sample size limit. The sample size limit is that it is 20 to 100. Number of sites, it is 1 to 5. Baseline adverse event rate is 0.20 to 0.80. Countries range is 1 to 10. Visits per patient range is 1 to 20. Dropout rate is 0.00 to 0.50, and recruitment rate is 1 to 50. Double blind, you need to select it or you need to remove it. Okay, so that is something you have to choose. Then oncology, here you can choose for which disease area you are performing this. And once you have selected, then you have to click on predict. It is going to predict you two things. One is the estimated cost. So the estimated cost here comes with 186,000 $176, and the risk is 96%. Very high risk is there. So this is how it calculates the cost, as well as it is giving the risk factor also. Both the things it is giving. Now, further, it is doing explanation also. It is giving sample size, phase, number of sites, then number of countries, then recruitment rate. So it is giving the factors which are affecting the most. These are the factors which are affecting the most. So it is giving those factors as well. Now going back here. Okay. Yeah. So the user interface is built using Streamlit. Okay. Now the key UI sections, sidebar inputs, dynamic inputs based on trial phase. Example, phase one. So phase one, small sample, there will be high AE rate. Okay. So we can see that, okay, we can make those changes. So sample size small, we are keeping 20. Okay. And rest we are keeping same and making the prediction. So you can see that 96% is the AE rate. Then you can increase the sample size. So I'm increasing it to maximum and then I'm making the prediction. So here you can see actually our model, when we have done this, so it is giving high AE risk rate if we are increasing the number of sample size. If we are keeping it near about 56, in this particular case, if I'm making the prediction, you can see it has got reduced by 2%. It has come back to 96%. So here we need to write the interpretations that how the increase or decrease in sample size is affecting the AE risk. At the same time, we should see this cost also, $186,176. So how the number of sites, okay, increasing the number of sites would affect. So here you can see it is making some change. So logically, I can comment that it is working on this sample data. So probably it is not working on very realistic way because this is sample data. If we increase the number of sites, obviously the estimated cost would also increase. Okay, so that's the bar which explains that we can choose the phases. Then we can click on the predict button, and when we click on the predict button, how the algorithm works, it prepares the input, match input format with training data, then load models. So joblib.load cost model.pkl, then predict. It basically gives cost underscore pred is equal to reg.predict. It makes the prediction. So these functions are called clf.predict proba. So these functions are called. So when these functions are called at that particular time, it brings those outputs here on the screen. So this is something it is bringing up here. Now, explainability is very important. So using SHAP explainable AI, so it shows that which features contributed most to cost. Top 5 drivers displayed as bar chart. So it shows these things. Example insights that how sample size is affecting the cost, how sites affecting the cost, how AE rate is affecting the risk. So that basically we can see. So here we can make changes. Let's say we have dropout rate is very high. So how it is affecting the cost? So cost is increased 187 for 56. If dropout rate is very low, in that particular case, you can see this time the cost has got increased more. Okay, so this is telling that how the dropout rate is actually okay. So at which rate the cost is optimal. So it is telling that. Once again, this particular model, okay, we cannot rely on this. It is not something to be used for real time. For that, we actually need to have real data here, and training on the real data. The model should get trained on that. Then we can expect on this, okay, the model will learn in a better way. Okay, then if we integrate LLM also, large language models as well, in that particular case, it will have Grok API integrated with a model Llama 3.3.70 billion versatile. So that model can be integrated, and it can give explanations for cost, for risk explanation, for optimization suggestions. So it can give all of these explanations in that particular case. Then we also need to work on the model performance, for which we can use the regression metrics and RMSE metrics. So metrics, they tell us that how the model is performing. Regression metrics examples are RMSE, which tells error, and R square, which tells accuracy. And classification metrics, so we can have ROC AUC curve, which tells that how well the model is behaving or well the model is performing. Here we have a tech stack used. The tech stack is from requirements.txt. We can see that what are the technologies used in the project. So here we have a file called as requirements.txt. So it has got Streamlit, Pandas, scikit-learn, XGBoot, Boost, LightGBM, SHAP, Matplotlib, Joblib, Grok, and Python .env. So these are the technologies which are used in the project, which we can see here in which we can see here in the requirements.txt file. So the end-to-end workflow is we have data set, pre-processing of the data, model training, save the models, get the user input, prediction, and then explainability and insights. So this is the end-to-end workflow of this project. If you need, you can ping us. We will share this project with you. So you can configure and try and probably enhance this project. The real world value this project can be used by, so it can be used by pharma companies, it can be used by CROs, contract research organizations, clinical researchers, and it will help in reduce the trial cost, also help in improve the drug safety, help in faster decision making, and also explainable AI, which will make it more regulatory friendly. The limitations of this project are: it uses synthetic or simplified assumptions, it is not validated for FDA or EMA use. It needs real world clinical data validation. The project structure, if you technically want to understand, then you can go through this. That it has got a root folder, which is ctdd, and then it has got some key subfolders: lib, scripts, include, etc, share, py, vnv for the virtual environment, and then like what is the purpose. So .env stores environment files, then model files are kept as ae model pkl, cost model pkl, clinical trial cleaned csv file, this is the data set. App.py, it is the main application file where all the logics are written, and requirement.txt is the file where all the required files are written, libraries or dependencies are written, and like that is how the project structure is organized. Here you can see this is the project structure on the left hand side. Okay, fine. Now moving ahead. So this was it, and here in this document we have explained that how when you change some parameters, how they are affecting. So you can also change and keep on having experience that when you change a parameter, how does it affect on the outputs. Further moving, let's go back. Okay, let me close this as well. Fine. So now let us try to see. So quickly we will walk through this the further chapters so that you can get further idea as well. So data sources for clinical trial. Okay. So quality and diversity of the data determines the ceiling of your model's performance. Experienced clinical data scientists know their data sources deeply, their structure, their limitations, even the biases. You can go for clinicaltrials.gov. So yeah, so from here you can choose. Okay, you can select. So for current location, probably they want. Yeah, so this one we, for current location probably it won't give. So allow this time. Yeah. So Location I'll have to, okay, update. No, it says our country. Okay, fine. So, zip code, state, it's not taking up. Okay, I've just entered. Apply filters. Okay, only one record you can see here. Okay, so from here, we need to look for more filters and put more filters and get the desired data from here. So this is one of the sources which are there for data purpose. Okay, getting the data. So what it contains, it is world's largest clinical study registry having 450,000 plus registered trials. Key data elements include trial identifiers and CT number, sponsor and phase information, therapeutic area, condition, mesh terms, intervention, interventions, drug name, dose route, inclusion, exclusion criteria, free text, primary and secondary endpoints, trial status, recruiting, completed, terminated, results data for completed trials, enrollment, outcomes, adverse events. So AI uses with clinical trials data we have are analyzing historical protocol designs to inform new trial design, benchmarking competitive trial landscapes, training models to predict trial success and failure, identifying enrollment benchmarks by therapeutic area. The limitations are results are self-reported and not always complete. Data quality varies, and terminated trials often lack posted results. Then this is one clinical trial data. Second is EHR, electronic health records, where you can work. So it contains longitudinal patient records from clinical care system. Major EHR vendors include Epic, Cerner, Oracle Health, Meditech, Allscripts. So EHR data types, structured data, diagnosis, ICD-10 codes, procedures, CPT codes, medication, NDC codes, lab results, LOINC codes, vitals. Then unstructured data, you will find clinical notes by physician, nursing specialist, discharge summaries, radiology reports, pathology reports. Semi-structured, problem lists, medication reconciliation, and care plans. What are the AI use cases? Patient identification for trial recruitment, generating external control arms, real-world comparators, post-market safety surveillance, then predicting disease progression and prognosis. Key challenges are interoperability, data logged in different formats across health systems because there is no standardization as such. So that's one of the challenges. Then missingness, values missing, not at random, didn't get tested, not that value was normal. Okay, so that's another challenge. Then bias, data from academic medical centers may not reflect community practice. Then privacy, HIPAA in US, GDPR in Europe, de-identification is required. Then another type of data which we have is genomics and biomarker data. What it contains? Genomics, whole genome sequencing like WGS, whole genome sequencing WES, exome sequencing, SNP arrays, from biobanks like UK Biobank with 500,000 plus participants. Transcriptomics, gene expression data, RNA sequence, which genes are active in patient cell. Proteomics, protein expression profiles. Metabolomics, small molecule metabolite profile. Liquid biopsy, circulating circulating tumor DNA, ctDNA for cancer monitoring. The AI use cases with genomic biomarker data are biomarker discovery for patient stratification, companion diagnostic development, toxicity prediction, pharmacogenomics, identifying responder versus non-responder signatures. The key challenges are high dimensionality. A single WGS produces 3 billion data points, and feature selection is critical. Small sample size, many trials don't have sufficient genomic data for robust ML modeling. Then batch effects. Sample processed at different times, labs can show systematic differences also. Interpretation, connecting genomics variants to clinical outcomes require deep domain expertise. These are the challenges. Okay. Now, we will have next this chapter completed, and probably we will have only one more chapter, okay, to be discussed here. That we will see as per the time we will go ahead or not. Now, ML workflow for clinical trial analytics. There has to be a structured workflow. A structured ML workflow ensures reproducibility, regulatory defensibility, and scientific rigor. For pharma applications, this workflow must be documented in detail and validated, not just run interactively in a notebook. So, step number one, problem definition. Before touching data, define business question. What decision will this model inform? Example, which site should we activate? ML task type, classification, regression, clustering, NLP. We need to decide. Target variable, label. What are we predicting? How is it defined? Example, enrolled is greater than equal to 5 patient by month 6. Model output, score, binary prediction, ranking, text extraction. Then performance requirements. What accuracy, recall is needed for the decision to be actionable? Validation strategy. How will you validate the model before deployment? Then next part is data pre-processing. Clinical data is rarely analysis ready. Pre-processing is often 60 to 70% of the ML project effort. It could be missing data completely at random, safe to impute with mean medium mode. Missing data at random, impute using other correlated variables, multiple imputation. Missing not at random, requiring domain informed treatment, may need to model missingness itself. Techniques, KNN imputation, mice, multiple imputation by chained equations, model based imputation. Second thing is categorical encoding. One hot encoding for nominal variables like country, disease type, ordinal encoding for ordered categories like severity, mild, moderate, severe, target encoding for high cardinality categories like site ID with 200 levels. Then nominal normalization and standardization. We have min-max scaling techniques scales all features to 0 and 1 range. We have Z-score standardization centers around mean with unit standard deviation. Then log transformation, useful for right skewed variables common in clinical data, lab value drug exposure. Then we have date time features like extract trial duration, time since last visit, days on treatment, create rolling window for time series features. Step number three is feature engineering. Feature engineering is creating new informative variables from raw data. often derives the biggest improvements in model performance in clinical trial applications. Examples of engineered features: screen failure rate per site per therapeutic area from historical data, average days from IND to FPI for similar trials at this sponsor, patient comorbidity index, Charlson comorbidity index from ICD codes, number of competing trials recurring in same indication in the same geography, biomarkers change from baseline at an interim time point. The techniques which are used for the engineered features: domain knowledge, correlation analysis, variance inflation factor VIF, variance inflation factor, recursive feature elimination RFE, lasso regularization. Domain knowledge is most powerful source of good feature. Correlation analysis, it removes redundant features, Pearson correlation coefficient or Spearman. Variance inflation in factor, detect multicollinearity. Then recursive feature elimination, algorithmic feature selection. And then lasso, it is embedded feature selection during model training. Step number 4 is model training and evaluation. So training, validation, test split. 60-20-20 or 70-15-15 split could be there. 60% train, 20% validation, 20% test. 70 train, 15-15 validation and test split. For time series clinical data, always use temporal forward chaining cross validation. Train on past, test on future. Random splits create data leakage in longitudinal data. For small dataset common in rare disease trials, k-fold cross-validation, k could be 5 or 10. Evaluation metrics for classification are accuracy, precision, recall, F1 score, AUC-ROC, AUC-PR. For regression, we have RMSE, root mean squared error, MAE, mean absolute error, R square coefficient of determination. For hyperparameter tuning, we need we need grid search, which is exhaustive search over specified parameter grid. Random search, randomly sample parameter combinations, more efficient. Bayesian optimization, intelligent search using prior result to guide next experiment, best for expensive models. Step number 5 is model validation and deployment. Validation requirements for clinical trial AI beyond standard ML. So prospective validation, test model on truly future data. External validation, test on data from a different site, different sponsor, or a time period. Clinical validation, work with clinicians to confirm predictions make clinical sense. Regulatory documentation, model card, data lineage, version control, audit trail. Now, deployment considerations are model monitoring, like detect when a real world performance degrades, that is data drift is happening. Then retraining cadence, how often should the model be updated with new data. And then human in the loop for high stakes decision, like for safety purpose, AI should be decision support, not autonomous decision maker. Now next point is handling challenges. So imbalanced data sets, okay, data quality issues could be there. Now, there are two of the most common consequential issues in clinical trial ML applications. One is imbalanced data set. For example, we have more records for healthy than less record for the diseased. So let's say 500 records for healthy and 50 records for diseased. So this is imbalanced data. So why it is pervasive in clinical trials? Serious adverse events are rare. 1 to 5% of the patient may experience the serious adverse events. Trial failures versus success, depending on phase, class ratios can be 3 is to 1 to 9 is to 1. Rare disease trials may have only 50 to 200 patients total. So this is basically, you can see that why imbalanced data is getting generated. So consequences of ignoring imbalance, a naive model on a 95 is to 5 class imbalance, 5% SAE, serious adverse event cases are there, can achieve 95% accuracy by predicting the majority class every time. This is completely useless and potentially dangerous, because we are more focused on the serious adverse events which are rare, and they are getting ignored by the model. In this particular consequence, we could observe techniques for handling the imbalance. We have data level methods. So SMOT is there, ADASIN is there, random undersampling is there, cluster based oversampling is there. So SMOT is synthetic minority oversampling technique. So whatever is in minority, oversample it, create more synthetic minority class examples by interpolating between the real examples. Then ADASIN is there, adaptive variant of SMOT, generates more synthetic examples in harder to classify regions. Then random undersampling is there, remove majority class, okay, risk losing information that is there, but remove the majority class examples. Then cluster based oversampling is there, oversample within minority class clusters, so that you can do. Now algorithm methods are there, class weights, tell the algorithm to penalize misclassification of the minority class more heavily. So class weight balanced in scikit-learn, we can use class weights as well. Then threshold adjustments, move the decision threshold from zero point 0.5 to a value that balances the precision and recall for the specific use case. Then ensemble methods, balance bagging classifier, easy ensemble. Okay, so that can be there. Evaluation, don't use accuracy, use AUCPR, F1 or Matthew's correlation coefficient for imbalanced clinical data sets. Okay, that is useful. Now, 3.4.2 is bias and data quality issues. So, types of bias in clinical data. Selection bias is there, label bias is there, algorithmic bias is there. Selection bias, clinical trial population are not representative of the real patient population, typically younger, healthier, less diverse. Electronic health record data from academic centers over represents complex cases. Now, impact models trained on trial data may not generalize on the real to the real world problem. That is a challenge. Okay, then label bias, adverse events are underreported in spontaneous safety systems, estimated 10x underreporting for non-serious events. So that's another label bias. Then subjective clinical assessments, pain scores vary by investigator. Impact, the ground truth label in your training data may be systematically incorrect. Algorithmic bias, models may perform better for demographic groups than overrepresented in training data. A model trained predominantly on Western, predominantly Caucasian populations may perform poorly for Asian or African populations. Impact, AI in clinical trial must be evaluated for fairness across gender, age, race, and ethnicity subgroups. Data quality dimensions, completeness, what percent of the expected data is present, accuracy, is the data recorded, value, the true value, consistency, are the same concepts recorded the same way across sites, timeliness, is the data entered promptly after the event, validity, are the values within plausible clinical ranges. Okay, then practical data quality checks, range check, heart rate between 30 to 250 BPM, cross field consistency, female patient with prostate cancer diagnosis, data entry error could be there. Temporal logic, example study discontinuation date must be after enrollment date, duplication, duplicate record, detection should be there. Okay, so in the chapter 3, we discussed the concept supervised learning, deep learning, NLP, data sources, ML workflow, imbalanced data, and bias. So supervised learning, core technique for prediction like logistic regression, and tree ensemble for most common. Deep learning powerful for images and sequence, require data at large scale, and explainability is there. NLP unlocks 80% of clinical data that is unstructured. Data sources, clinicaltrials.gov, EHR, genomics, each with unique structure and limitation. Then ML workflow, problem defining, pre-processing, feature engineering, training, and evaluation. Then imbalance, smart class weights, AUCPR, don't use accuracy alone. Bias, selection bias, label, algorithmic bias, evaluate fairness across demographics and subgroups. So this is the summary of the chapter 3. Now, what are the remaining chapters which I would cover in a separate session? So remaining chapters are about predicting clinical trial outcomes and success rates. Then next chapter is yes, we can look at the chapter from here. Yeah. So you can see third we have covered. So chapter 4 is predicting clinical trial outcomes and success rates. So you can comment on YouTube whenever you are watching this video, what more you require from the previous chapters covered. And these chapters also we will cover in further because now we are almost hitting towards the time. So predicting clinical trials outcomes and success rates, the topics which come under under that is AI for trial success prediction, phase transition prediction, and then risk categories, safety risk, efficacy risk, and operational risk. Then predicting modeling techniques like binary classification, success failure, survival analysis, improving decision making using predictive insights. Next chapter, fifth, is AI for drug safety and pharmacovigilance. And under this, we have introduction to pharmacovigilance, adverse drug events, and serious adverse drug events. AI techniques are NLP for safety reports, signal detection using ML, and then real-time monitoring, variables and patient-reported data, automated alert systems, predicting toxicity and severity for adverse events, case study, early detection of safety signals. Chapter 6 is AI across clinical trial phases. So we will see for all the phases, phase 1, 2, 3, 4, and then AI adaptive clinical trials and simulation-based trials and digital twins. Chapter 7 is regulatory requirements and compliance. So we will go through the global regulatory frameworks, FDA from USA, EMA from Europe, CDSCO from India, key compliance standards, GXP good practices, 21 CFR Part 11, Part 11, AI in regulatory submissions, data traceability, audit trails, ethic considerations, patient privacy like GDPR, HIPAA, and bias and fairness in AI. Then we will go for chapter 8, that is explainable AI, that is XAI in healthcare. Under it, why explainability is critical in pharma, black box versus interpretable models. And then we will go for techniques like SHAP, LIME, model transparency for regulatory approvals, building trust among clinicians and regulators. Chapter 9 is hands-on project and prototype model and dashboard. So today we saw one of the prototype, and in the next sessions, I would explain more of the project and prototypes. Then uh, predicting clinical trial success rate, adverse event, data set preparation, small clinical data set, model building with logistic regression and random forest, evaluation metrics, accuracy, precision, recall, and F1 score, dashboard creation, Power BI, Tableau, insights generation, risk factor, cost saving opportunities. Chapter 10 is strategy and implementation. And here, implementation in pharma organizations, how does that happen? So here we will be covering AI adoption roadmap, pilot scale enterprise deployment, integration with existing clinical work, building cross-functional teams, data scientists, clinicians, regulatory experts, ROI measurement, cost reduction, faster time to market, real world case studies. And the last chapter would be career pathways in AI driven pharma, where we'll be covering that what are the different roles: clinical data scientist, pharmacovigilance analyst, AI healthcare consultant, and the skills which are required: SQL, Python, ML, BI tools, certifications and learning roadmaps, and industry demand future trends. So these we will be covering up in the next session. I would like to thank everyone for taking out so much of your time and is spending Sunday on learning uh, how AI is affecting the clinical trial and drug safety. Once again, thank you very much. And share this video with all of your friends and those who need, and inform them that we will have more session on this. So, and to get connected with us, you can directly comment on the video. Okay, if you need some more topics, which topics would you like to cover in the area of pharmacy, artificial intelligence, drug discovery? You can mention. We will come up with those topics. Thanks a lot. Thanks for watching, and don't forget to subscribe.